Hello everyone, welcome to Dr. Osman's Edu Trends, swinging to educational trends. Today we'll be talking about rational drug designing in medicinal chemistry. So we have to first talk about the word rational. So what do you mean by rational? Rational means unique or agreeable, like anything which is unique from the thing which is already with us before, like something which is very much unique in its properties or in its qualities, or if we are talking about a drug, then we need to see that it is unique in its potency, it is unique in its dosage, like if you are taking 10 mg of drug, if it is producing some action, then the new drug, the rational drug, should produce more action or in less amount of drug, or like a 5 mg if you are taking, then it should produce the same action or more action, and even it should have less side effects than compared to the drug which was already existing, right, which is with us only already before. So we need to go with the rational drug designing, that the designing which is actually unique than compared to before. Now if we talk about the definition of rational drug designing, then we can say that it should involve logical effort. Logical means it should have a logic actually for discovery of new chemical substance with maximum potency, right? The potency should be more, because every time potency means the pharmacological action. Efficacy, it should be effective. Safety, it should be safe, and minimum drawback. Now if we go some more inside this, we should understand that there should be a logical sequence of efforts when we are discovering a new chemical substance. Then whatever drug we are getting, it should have more potency, more pharmacological action, the drug should be effective, right? Like whatever drug we are taking, that much amount of drug should be reaching the site of action. Sometimes if you take some amount of drug and the same quantity of drug is not reaching at the site of action, means the action will not be proper, like we won't get the desired pharmacological action. So that's why it should be effective. Then safety means the drug should be safe, means the drug should not produce other metabolic waste or toxic product. If it is producing other metabolic waste or toxic products, then there will be a disruption actually, there will be no proper treatment. And minimum drawbacks, this means like there should be no side effects or adverse effects. Like generally it happens, sometimes whenever we are taking a particular drug, you will have some side effects. You may have some giddiness, you may have some stomach pain, you may have some headache or something, but the side effect should not be there. But this is allopathy medicine, we are talking about the medicines which comes under the allopathy medical science. So here side effects will definitely be there, but our job is to synthesize, or our job is to discover a particular chemical substance that should have less side effects. If less side effects will be there, definitely there will be more potency. Now if we talk about the purpose, like why we want to go with rational drug designing. Rational drug designing is done to synthesize a drug which possesses greater chemotherapeutic index and specific action. Chemotherapeutic index, specifically the bioavailability, right, like what I said you just before, the amount of drug which is available at the site of action. If that much amount of drug is not available at the site of action, then definitely there will be no pharmacological action. So the drug should be properly available at the site of action, and the action should be specific, right, at the specific site of action. Now, so rather we can say that rational drug designing is a program research for an ideal drug. It is a program research, it is a planned research, and it is preferred to avoid the synthesis of thousands of drugs which are useless, which are not actually come into practice, like we cannot use them further because of their drawbacks. Now also rational drug designing involves the study of various biochemical reactions, like you all are aware that whenever we take something in our body, that particular foodstuff gets mixed up with lots of chemical which are already inside our body, chemicals in the forms of enzymes, or you can say so many secretions inside our body, that actually gets mixed with that, and lots of biochemical reactions goes on. Example, I can say you medicinal biochemistry, when we are taking some amount of carbohydrate, or when we are taking some amount of fat, or when we are taking some amount of protein. So what happens? That protein, that carbohydrate will get interacted with the secretions of your mouth, and then whatever enzymes present there will interact with that particular foodstuff, the biomolecule, and it will try to change it so that the body can easily absorb that biomolecule. So series of biochemical reactions goes on inside the body. Then whenever the drug is going inside the body, when it is getting absorbed, and again when it is distributed and reaching the site of action, there will be drug molecular interactions. So that, being studied in this program research, that is rational drug designing. There are so many chemicals which are again synthesized as triggers. Whenever some reaction is start happening, then again some drug, it gets synthesized automatically inside the body. It also involves the study of spectrum of activity, like the drug which we synthesize, or the drug got synthesized with the help of rational drug designing. The drug is able to kill the bacteria of different types, gram positive, gram negative, or narrow spectrum, or broad spectrum, or the drug is able to kill both the bacteria, or the drug is able to kill the fungi, protozoans, so many microorganisms which are actually the reasons for the existence of disease. And later, if the toxins are produced, the metabolic products are formed, then we need to study that also. Then that studies are called as toxicological studies. Then many biotransformation reactions goes on, like biotechnology, rather we can say phase 1 and phase 2 glucarinoid formation and all, that also being studied, and metabolic studies, as I told you, etc. Now we have some examples of drugs which are developed by rational drug designing, like hypoglycemic agents. We have insulin analogs, we have somatostatin analogs, we have fatty acid oxidation inhibitors, we have anorectic agents, example mazindol, cyclisation. We have aldose reductase inhibitors. Okay, so they are all the examples. Then we have anticoagulants like the vitamin K. It is cyclized to give a different form actually, so that the activity or the mechanism of action or the potency of the drug is enhanced or improved. We have antimetabolites, example pyrimethamine and cycloguanil, which is made with the rational drug designing process. Now let us talk about lead. We say it as lead as it is actually a prototype compound which is also called as the basic nucleus, and which possess desired pharmacological or biological activity along with few undesirable characteristics. Now we heard this same spelling word as lead in periodic classification as the symbol Pb, but here we call it as lead because lead is nothing but is something to take forward, and this basic nucleus of structure, the basic nucleus of the structure, or the basic a particular site, for example, if we take an atom, it will have nucleus, and nucleus is basic nucleus, right? It will have all the genetic material inside it. So here also in structure we have a basic nucleus, that is lead actually, and it is actually prototype compound. It is actually the main compound which will have the complete mechanism of action. Particularly, for example, if you say penicillin will have a basic nucleus, and if you do anything to that basic nucleus, and anything is done on the basic nucleus, or anything to change, so it will affect the main biological activity, it will affect the main pharmacological activity, that is antibiotic nature of penicillin. So here also we need to identify lead compound, and the identification of lead compound is the heart of drug designing. So first of all we need to identify a lead. How we can do that? We can do the identification of lead nucleus by taking natural substances which is available in the nature, and that can be done by discovering that can be done by various processes like random screening, non-random screening, drug metabolite studies, clinical observations, and rational approaches. Rational approaches like for example the site of mechanism of action at cellular level, molecular level, as well as the electronic level of that particular atom. Then we also need to study the 3D structure of receptor and its complementary with that of the drug to be designed.
So it is very, significant in the identification of lead nucleus if we go on studying the above said, activities, or if you do, and if you go on studying the above said, things. So lead discovery actually involves a planned work which includes signs and symptoms of concerned disease against which the drug molecule has to be prepared. Now we all are aware that whenever we need particular drug, we actually don't go with the discovery of drug first. We first get the disease in existence. For example, we have plenty of diseases in the universe, plenty of microorganisms and plenty of diseases. So first of all, disease will get notified and we will start preparing a drug for that disease. So first we will see how the disease is caused, what is the pathology of that disease, what is happening actually, how the physiological functioning of the body got affected, and how that particular microorganism has affected that particular part. On the deeper level, we will try to study everything, and then again we start discovering a drug which is specifically beneficial in treatment of that particular disease. So first studying the disease is a important. Then after studying the disease, we all will go on planning a drug that is particularly effective in such kind of disease. Now after identifying a particular lead, we need to see that particular lead nucleus is according to our need or not. Sometimes what happens that lead nucleus is not particularly with our requirement. So first of all, we need to modify that lead nucleus. We have to change something in that lead nucleus so that it comes into use of our desired process of making a new drug. So we call a word like optimization. So what do you mean by optimization? You all have heard a word called optimum temperature. That means temperature, at particular temperature this should be done like that. So optimization of lead is nothing but we are optimizing it. We are actually trying to make it palatable or make it into use. So optimization, another example I can give is the regulator of a fan. So if you want a fan in full speed, especially in summer, so what we do, we just switch the fan on the higher speed, and sometimes we want less speed or less air, so we just go with the lower speed. So we actually try to regulate, we actually try to optimize the speed of fan so that we get the desired aeration, right? So we don't want every time more, we don't want every time less, so we just regulate the speed. The regulation is nothing but optimization. So we are optimizing the lead. So after the identification of lead, it is to be refined by various methods in order to develop the pharmacological properties of that particular drug as per our need. So first step in this is identification of pharmacophore. So what's happening here? Pharmacophore is an essential part of drug molecule. Pharmacophore is actually that particular area or that particular part of that chemical structure which actually possesses or exerts pharmacodynamic properties, or you can say the action, the pharmacological action or function. So pharmacophore, rather we can say it as a schematic representation of an active group along with interatomic distances, and the geometric arrangement of pharmacophore is called as pharmacophoric pattern. Now after identification of pharmacophore, structural modifications are undertaken to improve the pharmacokinetic properties of the drug. So we just go on improving the structural modifications to improve the pharmacokinetic properties. What are pharmacokinetic properties? Absorption, distribution, metabolism and excretion. So absorption is important. To improve absorption, we need to see solubility, we need to see partition coefficient, we need to see dissolution, we need to see disintegration, okay, so that the absorption is proper. Then again after absorption, metabolism. Then after metabolism, there should be good distribution, like that. So if we do anything in the structural modification, the pharmacokinetic properties will be affected. Now we have an example of morphine, which is an analgesic agent, narcotic analgesis. So here this morphine causes respiratory depression and sedation, and this is actually a side effect, and this can be minimized by identifying pharmacophore and modification. So we have the structure of basic nucleus of morphine, and you can see that we get three compounds if we are just changing particular atom at a particular place. So you can see in the structure, when N-methyl morphinans, they are obtained by removal of tetrahydrofuran ring and hydroxyl group at R and R1. So you can see when we are replacing H with methyl, right, or acetoxy, then we are getting different compounds and we will be suppressing the adverse effects of that particular nucleus. Now if you move on to the second subclass or second substep, optimization of functional group. Now in first step what we did, we actually identified the pharmacophore. In second step, we are just optimizing the functional group. Now we are talking about the functional group which is attached. The functional group can be anything. It can be alcohol, it can be aldehyde, it can be ester, it can be carboxylic acid, right? So we have to again see that the functional group which is attached, again it should be according to our need. The drug action here is correlated with functional groups, right? And again that functional groups again will be related to pharmacokinetics like lipophilicity, electronic and steric parameters, etc. So here functional group selection must be opted to avoid side effects and to control drug distribution pattern. Now again we have a example here. We have carbutamide and tolbutamide. So carbutamide is actually an antibacterial drug and it can be converted to antidiabetic tolbutamide simply by replacing NH3 group with methyl group. So you can see the complete activity of the drug is getting changed only by replacing a particular functional group. So you can see how much it is important to understand that a simple functional group can completely change the activity of that particular compound. Now moving on to structural activity relationship studies. Now chemical constitution, it is important here and it is a base to study SAR, that is structural activity relationship. Now basically if you want to study structural activity relationship, I advise you to take a example of bike. Bike actually it's bi. Bi means two cycles. So we say it as a bicycle. Bicycle which is manually can be operated with the help of pedals. Now if you are attaching engine there and if you are providing a battery and if the battery is providing the electricity, the engine will be getting activated and it is making that bike run with the help of that engine. So the rear wheel will start rotating and it will take the complete bicycle or bike along with it. Now have you ever seen anything which is not a bike? For example, two wheels are always called as a bike. You cannot call three wheels as three cycles as three wheels as three-wheeler. You cannot call three-wheeler as a bike. Why? Because two wheels are actually two cycles. So bicycle. Now if you want to change the seating arrangements of that particular bicycle, you can attach a big seating arrangement. You can change the handle. You can change the speed of that particular engine, the efficiency of the engine. You can do many modifications there according to your need, but the basic structure is same. The only two wheels and everything is changing about them according to your need, but the bike is not changing. The bike what is not changing? You will get different bikes or different companies. You can see every bike will have its own efficiency. You will get a bike which can give you good mileage in 1 liter of petrol. You can get another bike will give you less mileage in 1 liter of petrol, but seating arrangement is different, efficiency is different, speed is different. So what I want to say is, by taking this example, you can understand that if you're changing anything on the structure, the activity of that particular thing will change. So here also, when chemical structure is there, if you're changing anything on the chemical structure, if you're doing anything to functional groups, if you're doing anything to the atoms, if you're doing anything to the elements, the complete activity of the drug is changing. You can see, you can even get reminded of the above example what I told you, carbutamide and tolbutamide. The complete activity of the drug is changing just by replacement of a single functional group. Now here, if you want to study structural activity relationship, two types of drugs are available based on the specificity with respect to structure. Structurally specific drugs and structurally non-specific drugs. Structurally specific drugs, these drugs are actually binds to a specific site like receptor, enzyme, DNA, etc. And structurally non-specific drugs will have no specific site to bind with in order to exert its pharmacological action, such as gaseous anesthetics or disinfectant and most of the sedative hypnotics. Okay, so these two actually has a different binding nature. So first we'll have specific binding targets, whereas the second category is having no specific binding targets. Now structural activity relationship also involves synthesis of number of analogs and the structural activity determination is done until we get the desired analog. We have an example again here, sulfonamide, which exhibits antidiabetic, antidiuretic and antimicrobial activity. This compound has three activities. It can be an antidiabetic, it can be used in diabetes mellitus, it can be used as diuretic, it can be used to promote, urine production, increase promote in urine production, and it can be used against the microorganisms, that is antimicrobial activity or antibiotic. Now if you can see, the amino and sulfonyl group of this particular nucleus should always be in para position. So sulfonamide group may or may not be substituted, that can be done, but amino and sulfonyl group in this particular nucleus should always be in para position. It cannot be on ortho or meta position, it should be always in para position only. And the replacement of benzene ring inside with other rings, like benzene with other rings, furan, you can say, naphthalene, anthracene, or substantially, or replacement or substitution, replacement of benzene ring with other rings or substitution of benzene ring with other aromatic rings will decrease or abolish the activity of the drug. The complete activity of the drug will be abolished. Either it could be antidiabetic, either it could be diuretic, either it could be that antibiotic activity. So now if you are just going on substituting benzene ring with hetero aromatic substances, the potency of the drug can be enhanced. So what our intention is, we should get more potent compound. Now N substitution, that results in inactive compound. So if you cannot replace that N, now if we just replace dissolve, then we will get the following compounds. You can see here, antidiabetic compound, we have hydrochlorothiazide. So that we got with the replacement what I told you. Now talking about the fourth subtopic, that is substituents. So if we need to talk about the substituents also. When we are optimizing a lead nucleus, the substituents which are attached to the basic structure, and when they are attached in chain, that is when they are attached chain, we call it as homologation. So when we are attaching the substituent, we need to talk about the polarity, we need to talk about the partition coefficient, we need to talk about the electronic and steric parameter. So they are very important for a molecule and they are depending on the substituents attached. Now homologous series or homologation, if we define, it is an alteration by addition number of substance by adding number of atoms in the chain, and if you are increase in the side chain, it will increase or it will enhance the pharmacological property, but to some extent. And lipophilicity causes increases in activity, and if the solubility of the compound is less in water, definitely the drug will not be dissolved, definitely the drug will not be absorbed properly, and it cannot be reaching the site of action. So less solubility will decrease the activity, more lipophilicity increases the activity. The nature of action of drug changes based upon the different side chain substituents. So side chain substituents are very important here, that will change the nature of action. Example is promethazine, which is H1 antihistamic. It possesses 2-methyl group between cyclic ring and nitrogen present in the side chain. So promethazine with methyl groups between ring and side chain nitrogen exhibits tranquilizing property. Now we have the last subheading, that is cyclisation of optimization of lead nucleus. So cyclisation, you can say that when we are attaching two rings to each other, we call it as cyclisation, because we are attaching two rings. Cycles are getting attached, right? So in order to enhance the potency and efficacy of the drug, you can transform an alkyl side chain to an cyclic analog, right? So here transformation of alkyl side chain to cyclic analog is happening. Substituents which increase the binding interaction have to be incorporated. So we need to incorporate such substituents which are increasing the binding interaction. The drug has to be binded to receptors or enzymes and all. Here example is substitution of N-methyl group of chlorpromazine into closed piperazine ring will enhance the antiemetic action of prochloroperazine. So prochloroperazine is an antiemetic drug, so we are enhancing the property of prochloroperazine by substituting N-methyl group of chlorpromazine into closed piperazine nucleus. So this completes rational drug design. Thank you.